Hello, everyone. So this week, what we're going to talk in this screencast is how you're going to be extracting the genomic DNA from those bacteria which you have grown on week 1, you have harvested last week, so on week 2, and now it's time to get the genomic DNA out before you then quantify how much you get next week. So how do you do that? Well, first we have to understand the challenge that you have. So basically what you want right now is just the genomic DNA, just this. But this DNA is actually contained inside some organisms which are fairly complex. They're simple organisms compared to humans, stuff like that, but they're still fairly complex. Organisms that consist of different parts, like the cell wall, you have the cell membrane, then they contain so many other molecules, proteins, lipids, RNA, and stuff like that. So how do you get these molecules out? Trying to do as little steps as possible. Obviously, you don't want to deal with every single type of molecule one step at a time. And so you do that in 3 simple payments. Now, 3 simple steps where first you deal with the outside part. So the cell wall and the membrane here. You just get rid of this. And then once you release the DNA, you actually try to remove from the DNA also those proteins that still stick through electrostatic interactions with the DNA itself. And then finally, you separate the DNA from all the other components. So how do you do it, practically speaking? Well, 3 different steps. Let's look at the steps right here. So first part, how do you get rid of the cell wall and the cell membrane? So here are your cells that you have collected last week. They're now frozen. We'll throw them back. Now, the very first step you have to do is just transfer it into this glass type of bottle here. It's called a glass stoppered bottle. When you do this, you have to make sure that you actually record the volume, okay, with that pipette. Try to be as accurate as you can. Do not forget. This is important for the next steps later on when it comes to calculate the volume that you need of a certain reagent. So once you have transferred them there, first thing you do, you add 1 milliliter of this enzyme called lysozyme. This enzyme degrades, works basically, breaks down the beta-1,4 glycosidic bonds between a certain type of carbohydrates in the cell wall. And so when you incubate at 37 degrees for 15 minutes with this enzyme, lysozyme, you're actually basically degrading the cell wall of the bacteria. So now the cell membrane is exposed. It's time to deal with that. And you do that by adding an additional 2 milliliters of 20% SDS. This is a detergent, not too far off from the one that you use to wash your hands with. You incubate at 60 degrees for 15 minutes. And this detergent, since it's hydrophobic, what it does, it interacts with the phospholipids in the cell membrane and therefore basically degrades the cell membrane. Not only that, but also with the nature proteins by basically getting them to expose those hydrophobic amino acids which are normally inside. By doing that, these proteins basically they interact and they lose their secondary and tertiary structure or even quaternary structures if they have it. Last, third thing that SDS does, also it breaks some of the DNA protein interaction exactly because it denatures some of these proteins. But it does not break all of the interactions. So now you get rid of the cell wall, you lyse the cell membrane, so everything, the entire content of the cell gets released into your solution, into your buffer. Now you still have proteins which are connected, so they're attached to DNA through electrostatic interaction, so through opposite charges. And so the way you deal with that is just by increasing the concentration of sodium chloride in the solution to 1 molar. Notice that I've said increase the concentration of sodium chloride. Because if you remember, last week when you made your buffer, you actually already had some sodium chloride, a 0.15 molar concentration. So what you have to do now in this step is weight a certain mass of sodium chloride in a way that you bring this concentration to 1 molar. So how would you do that? I leave it up to you. So now you know the volume. If you remember, you measure the volume, and that's why it was important to do that, of the cells that you had at the beginning. You know how much lysozyme and SDS you added, so now you can know the total volume you have in there. Now you have also to take into account that the original buffer, so the volume of the cells that you had at the beginning, already had 0.15 molar sodium chloride. So how much sodium chloride in the powder form do you have to add now to get this entire volume to 1 molar? We'll do this calculation, we'll discuss them in class as well. So you add the sodium chloride, now you mix it and you let it go to room temperature. Now the sodium ions and the chloride ions will actually interact with the basic proteins and the DNA, but basically neutralizing these charges which otherwise would cause the DNA to interact with the basic proteins. By doing that, the result is that the proteins dissociate from the DNA. Great. So now you have your DNA just being finally separated from the proteins. You have your proteins. In the solution, you have your phospholipids attached to SDS, and so on. Now it's time to actually separate the DNA from all the other components. And you do that by using this mixture 24:1 of chloroform and isoamyl alcohol. You use an equal volume of what you already have in the glass bottle. So you add to it. Now, what the chloroform does, basically, it degrades or denatures the proteins in the solution. And the function of isoamyl is simply to stop the foaming from happening in this case. So what you do, you add an equal volume of chloroform and isoamyl alcohol. And then you add it to this and you shake for about 10 minutes. Okay, this is done at room temperature. Now, chloroform, this mixture is actually toxic, so this step has to be done in the fume hood. Make sure the fume hood is on. Okay, so now you can mix it outside of the fume hood, but then from time to time, since you have to shake it for about 10 minutes on and off, make sure that when you open the top, the stopper of the glass container here, you are inside the fume hoods. Okay, this will help to release the pressure. After the 10 minutes, now you're actually ready to separate the DNA by centrifuging. So you transfer the whole mixture from the glass stopper bottle here to the centrifuge tube, like the one you used last week, and then you spin the whole thing for 15,000 RPM, revolution per minute, for 10 minutes at 4 degrees, using the same type of rotor that you used last week. You'll do this step all by yourself. Okay, I will not help you with this. Remember how you used the centrifuge last week. So once you're done with your centrifugation, you'll have basically 3 layers. You have an organic layer here with chloroform and isoamyl alcohol here, an intermediate layer here, and an aqueous layer here. If you read your manual, you'll know where DNA is supposed to be, okay, which layer. Now, you take this layer, the layer, whatever layer you choose, you take it and you transfer it into a beaker. And now it's time to actually, so that layer will contain DNA, which is fairly pure, it's been fairly purified from the lipids, from the proteins, and stuff like that. Now, don't forget one thing is to immediately dispose the chloroform, because chloroform can actually melt the plastic, the polypropylene in the bottle there. So pour it out immediately and rinse this bottle as soon as you can. Now you have your DNA in the beaker, the small beaker. It's time to precipitate it. And what you have to do, you have to add 67% ethanol to this. So you take pure ethanol and you add it to this mixture in a way that it gets to 67%. How can you do this calculation? Think about it, and eventually we'll discuss that in class. Once you add the ethanol, what happens is that the aqueous solution that the DNA is in, because of the ethanol, becomes less polar, and therefore DNA has a hard time to stay in solution. DNA is a polar molecule, so will tend to precipitate, and so it starts forming like this filamentous structure right here, which then you can collect using a glass rod. You go around like what you do with what is it called, a candy, sugar candy cane, stuff like that. It's called candy cane? No, it's called cotton candy, sorry. What you do with cotton candy. Same thing, you go around, you slightly rotate. When you go around, you slightly rotate the glass rod too, and then basically the DNA will get stuck around. So if this is your glass rod, the DNA will get stuck all around here, okay, wrapped around the glass rod. Now you let it air dry, so you put it on the bench, make sure that the DNA, the part of the glass rod with the DNA does not touch the bench, so it's suspended. You let it air dry for 10 minutes. What happens when you air dry is that the ethanol will actually evaporate. Ethanol evaporates faster than water. So in 10 minutes you get basically all the ethanol that was in the DNA to evaporate, but the water will still be there. Okay. So you take it and then you resuspend the DNA from the glass rod in 10 milliliters of the buffer that you had from the week before. And so you do that and you collect your DNA inside the test tube when you do that. Then you give me this DNA and we'll store it until next week so that you can actually calculate what is the concentration of the DNA that you have achieved during all these purification steps. Okay, that's it, and I'll see you in class.